Chapter 21 of our textbook is going to cover those processes of genetic inheritance. So we're going to be looking at genetics and predicting the traits of offspring based on what we know about the genetics of the two parents. We're going to be doing that by looking at sections 1, 2, 4, and 5. So make note that we're not going to be covering section 3 as part of this class. Lots and lots of questions answered by this chapter. I'm not going to read them because I want to make sure to cover everything. So if you need to pause here, you can do that. So genetics is the study of heredity and the variation of inherited characteristics. In the study of genetics, heredity is the passing of genetic material and then all of the associated traits that go with them from the parent to the offspring. So we're going to look at how you get the combination of genes that you inherit from your parents and then describe those principles of inheritance. Inheritance just meaning that we are receiving those genes both from our mother and our father. And it's very, very common for situations to arise where the offspring either doesn't look much like either the mother or the father, or might have medical conditions that neither the mother or the father displayed. And those are all going to be explained by these different processes of dominant and recessive genes that we see during this process of inheritance. I myself am a blonde-haired, blue-eyed person, and both of my parents and my sibling are kind of olive skin complected. So I used to tell people, when I was young and kind of didn't know anything about anything, that I wasn't related to them. But through the study of genetics, I was able to see like how those characteristics are hidden in the genetics of both my mother and my father, but in me, because of the combination of genes that I inherited from them, they become the dominant traits in me. So let's start out with some really important vocabulary that we're going to need to really be able to define as we go to understand some of these concepts and then also to solve the set of problems that you're going to see in your lab for this week. So first one we've seen before, a gene, which is the segment of DNA that directs the synthesis of a specific protein. So a gene could code for hair, skin color, nose size, ear size, eye color, that kind of thing. All of the proteins that we've talked about that are making up all of the tissues within our cells, all of those are coded for by specific genes. Then we have the trait, which is the characteristic that is expressed as a result of that protein, as a result of that gene. A new vocabulary term that we might not know yet is allele, and these are different versions of the same gene. So we know by looking at our population that there is variation in characteristics, variation in skin color, skin tone, complexion, the tendency to, some people are allergic to bees and some people are not. All of that exists because we have different versions of the same gene floating around in our population. So they code for the same thing, but the trait that they express might be different. And within those alleles, some alleles are considered dominant, where if you have a copy of that allele, it will be expressed over any other copy of that allele. And then we have recessive genes, where if you have a dominant gene and a recessive gene, you're only going to see the trait expressed of that dominant gene. But if you have two copies of that recessive gene, that less strong one, then you will see that one expressed. So we'll see examples of all of that. More new terms, genotype versus phenotype. So our genotype, we can think of gene being the operative part of that word there. Genotype is the set of alleles that a person possesses for that trait. So recall back to mitosis and meiosis, when we're doing cell division, we hopefully remember that each of us in each of our cells have two copies of each gene. one from our mom, one from our dad. And our genotype is just a list of the copies of those genes that we have. So which one did I get from mom? Which one did I get from dad? Genotype. Our phenotype is the observable trait that we're going to display. So as a result of the genotype, how do we look? How do we act? How does that function? And that function, or how that looks, is the phenotype, the observable trait. Genotypes can be either homozygous or heterozygous. If you are homozygous for a specific trait, then you have two copies of the same allele. Remember, homo meaning same. So two copies of the same allele. If you're heterozygous for a specific trait, then you have two different copies of that, two different alleles for that same trait. And that's going to really depend on how many alleles for a specific trait are floating around in our population, and that will change based on which trait we're looking at, but we're going to see some examples of that in this chapter. We call this the study of Mendelian genetics because much of what we know about it was from work done by this guy, Gregor Mendel. He was a monk during the 19th century, and he conducted experiments on pea plants by crossing plants with different traits and determining the characteristics of their offspring. He conducted all of his research before we knew anything about the structure of DNA and chromosomes, which is amazing. So he's drawing all of these conclusions before we even knew anything about things like genes or the code of DNA. But even without all of that information, he discovered the principles of inheritance anyway, and then further research has shown us why these principles actually occur. So Mendel observed that some of the traits that he was seeing in pea plants were similar to the traits he saw in the parent plants, but some were different. He wanted to know how this occurred and if it followed a predictable pattern. So as a scientist, of course, he developed a hypothesis. And his hypothesis was that some traits are inherited from one parent and some traits are inherited from the other parent. And of course, he designed an experiment to test this hypothesis. Mendel spent about 10 years conducting these inheritance studies, and he looked at 7 different features in peas, including height, flower color, seed color, and seed shape. And to do this, he first had to establish lines of pea seeds with 2 different forms of whatever feature he was looking at. So we'll use our short and tall as the example here. He started by growing pea plants that were tall for generations, so generation after generation after generation, until they only produced offspring that were tall. And after doing this, he was confident that those plants could only pass on the trait for tall. And then he did the same thing to create a line of pea plants that were only short and produced only offspring that were short. Next, and that in this image is going to be called the P generation, pure generation. Only going to pass on the trait for tall in these guys, only going to pass on the trait for short in these guys. The next step is that he bred the tall plants with the short plants and then observed how the traits were inherited. The result here was that some of the offspring were tall. All of the offspring in that first generation were tall. But then when he bred these plants from the F1 generation, so 2 of the pea plants from the F1 generation, which were all tall, he bred those together to form the F2 generation. And he observed that every single time he did this, no matter which trait he was looking at, about 75% of the offspring were tall and 25% were short. And then interestingly, when he conducted his experiment on all the other traits, he found that the percentage of offspring displaying those certain traits remained the same. So in flower color, 75% of the offspring were blue and 25% were pink, with the pure lines that were crossed. And these are both examples of one trait crosses. And Mendel developed something called the Punnett square, where we can use the Punnett square to predict the possible outcomes of a cross. And that's what we'll look at next. We're going to look at the rest of our examples in humans because obviously this is a human biology class. So the example your book uses is freckles. And the trait of having freckles is determined by a dominant allele, meaning that if you have one of the copies of that dominant allele, that gene will code for freckles. You will have freckles. That gene will be expressed because it's dominant over the others. So let's say that a woman who is homozygous dominant, homozygous meaning both of her alleles for that gene are the same, and dominant meaning that they will code for freckles. And she is going to have a child with a man who is homozygous recessive. Homozygous, again, meaning that both of his alleles are the same, but they're both recessive, meaning they will not code for freckles. And here's kind of our system of abbreviating for genetics. So our big F allele is dominant. Our little f allele is recessive. Dominant leading to freckles, recessive leading to no freckles. Mom is homozygous dominant, 2 copies of the dominant allele. Dad is homozygous recessive, 2 copies of that little f recessive allele. Then we can match these up in a Punnett square to help us determine what the probability is that their offspring will have freckles. That's the purpose of a Punnett square, to determine the probability that the offspring will have a specific trait, in this case freckles. So the first step to creating a Punnett square is to determine all of the possible gamete combinations that each parent can form. In this example, each parent can only form one type of gamete when it comes to freckles because they're both homozygous. So mom will put her eggs on one side of our square. Mom can only donate a big F dominant allele, and dad can only donate a little f recessive allele. So we're going to put mom's eggs on the side here and dad's sperm on the top. And then we're going to match them up. Big F from mom, little f from dad. Big F from mom, little f from dad in each of those squares. Big F from mom, little f from dad. In this case, 100%, 4 out of 4, of the offspring will have freckles. So there's a 100% probability that their offspring will have freckles because there is no chance that they will not receive that dominant allele because that's the only thing that mom can donate. So let's look at that if we were going to perform a different cross where now mom is homozygous. So she has a copy of the dominant allele, but also a copy of the recessive allele. And dad is also homozygous. He has a copy of the dominant allele, but also a copy of the little allele, which means he would have freckles, right? Got to change our image. Got freckles. Okay, so now the gamete combinations are different. Mom could donate both a big F dominant allele or a little f recessive allele. And dad could donate a big F dominant allele or a little f recessive allele. And that's going to make our resulting probabilities different than we saw in our last cross. So here we're going to get a big F from mom and a big F from dad. Here we're going to get a little f from mom and a big F from dad. In this square, we're going to get a big F from mom and a little f from dad. And here we're going to get a little f from mom and a little f from dad. Again, this is an example of a one-trait cross because we're only looking at a single characteristic. But we see here that we have a different genotypic ratio and a different phenotypic ratio. We have a 25% chance of being homozygous dominant, 2 copies of the dominant allele, a 25% chance of being homozygous recessive, 2 copies of the recessive allele, and a 50%, 2 out of 4, chance of being heterozygous, having 1 copy of each type of allele. So those are different probabilities. In the next video, we're going to look at how to determine probabilities of a 2 trait.